शेयर
सर शेयर नहीं हो रही स्क्रीन अभी। अब हुई है। यस सर।
what to do with this. Who can start the collaboration?
सर रिकॉर्ड हो रहा है।
So okay, anyway, let's start. So what is I mean or maybe we have to start in today.
सर कल तो हमने फॉस्फोलिपिड किया था।
Okay, yes, today we have studied about phospholipids. No, in addition to that, we have studied about proteins as well, not only phospholipids. So today we are going to talk about a little bit about tertiary structure, and our main topic would be quaternary structure. But
ओके।
here, because to give you some background, I will start from the tertiary structure. Okay, so here you have tertiary structure. All right.
यस सर।
So tertiary structure, as I have mentioned yesterday, that there are some additional bondings here. In case of primary structure, there was only peptide linkages, which was directly between the amino acids.
यस सर।
While in case of secondary structures, we have uh, observed some other bondings there. Most importantly, there the bonding was hydrogen bond.
यस सर।
Hydrogen bonding was actually present in case of secondary structure. In addition to that, you have alpha helix. That alpha helix is formed, and definitely that is combined because of this hydrogen bonding which is present there.
यस सर।
Similarly, we have beta plated sheets which were also, both of these structures are formed because of the combination or because of the bonding of the hydrogen there.
यस
Because there is a carboxylic bond, carboxylic group is there, and you have hydrogens. So there's a weak bonding which is present in case of secondary proteins. So now, yesterday I have added one thing that uh, there is a hydrogen bond. Yes, that is already present. That is also already present in case of secondary. So definitely it is also present here as well.
यस सर।
Additional bondings here, which are most important, that is ionic bond, right,
यस सर।
and peptide bond.
ओके सर।
All right. So disulfide bond.
ओके सर।
All right.
यस।
So now we are going towards a disulfide bond, which will be formed here. Disulfide bond.
यस सर।
All right. So because as it is a disulfide bond, so it means the sulfur is involved here. So it means this bond will form in the tertiary structure, specifically there uh, where it has a amino acid or amino acid that has sulfur in itself as an element in itself.
Okay, sir.
So definitely this sulfur should be present in the amino acid. That's why that amino acid will be able to form this sort of bonding.
Yes, sir.
So for this, there is a amino acid that is cysteine. Remember,
Okay, sir.
cysteine. It's an amino acid, and this amino acid contains sulfur. This bonding will be formed here. So this cysteine contains sulfur.
Okay,
So
sir.
it means this cysteine is able to form a disulfide linkages in the protein of any. Definitely cysteine is present in most of the proteins. It is a part of the protein, so specifically in tertiary structure it's going to form a main disulfide bond. It is the strongest bond. Must keep in mind this sulfur bond that I have shown you here. It is one of the strongest bonds.
Yes, sir.
So what it shows us that all these bondings which are present in case of proteins, they are giving a very specific structure, a very defined structure to the protein. And that's why we always say protein has a very specific structure, very specific shape even.
Yes, sir.
And for instance, we have an example of an egg, and that egg has a protein. Once you boil that egg, so after that, that will be denatured, that the shape of that protein will be changed. So
Yes, sir.
as a result, you cannot convert that protein back into its original form, because it is now after the denaturation that you got uh, in a boiled form of the egg. So in this case, this specific shape is very much important for the functioning of the protein. Once you will change this shape by any way, like you may increase the temperature, or you may increase the pH, or you may do some other thing, or you may increase any type of any external uh, force or external environment. If you change it, and that if that will not be optimum, so it would be a problem for the protein, and protein will not be able to perform its function. So now
Yes.
let's move on the next point, that is quaternary structure of protein. Quaternary structure of protein. Quaternary structure of protein. Right.
Yes, sir.
So quaternary structure is actually, it is formed from more than one tertiary structure. When they put together, and they may consist of two to more polypeptide chains, and they form a globular structure. Remember, this quaternary structure is itself is not any specific structure. This quaternary structure is actually formed from the tertiary structure.
Okay.
How? By the combination of two to more tertiary structures. And we have in our mind that how tertiary structure is formed, right?
Yes, sir.
When that structure more than 2 to 3 times, when that will combine together, that will form a quaternary structure. So
Okay, sir.
here you will have a very specific shape, that is globular shape, right?
Yes, sir.
So first we need to understand that how this quaternary structure is formed. This quaternary structure is formed by the combination of more than one tertiary structures. I can mention here by the by the combination of more than one tertiary structures.
Yes, sir.
You get?
Yes, sir.
Okay. So when these, as I mentioned yesterday, that it is possible that that may contain 2 to 3 uh, tertiary structures. Definitely, that's a polypeptide chain, so it is possible 2 to 3 polypeptide chains are actually involved there. All right. So they will form a structure that is called globular structure. It is important here. Globular, right? Combine. Globular structure. How this structure is formed? This structure is formed by the combination of 2 to more tertiary structures. And this globular structure is very much important in case of protein, which is going to perform any specific function. And we will get some examples. For example, there is a hemoglobin. Hemoglobin. It is a globular structure, and we
Ye-
will see if the globular structure of this hemoglobin will change. So as a result, it will not be able to perform its function, right? And
ye-
there will be a specific disease that we will discuss very soon. Okay, so
ye-
any structure which is not made up of amino acids but is the part of protein, that is called prosthetic group.
Okay, sir.
Prosthetic group. I'm writing here. Uh, sorry. Prosthetic group. You will also study this term in case of enzymes as well, and there you will also study that it is non-protein part of uh, the enzyme,
Okay, sir.
because enzymes are also proteins.
Yes.
So this is a prosthetic group which is a non-protein part. For example, what would be this here? Heme group. In case of hemoglobin, heme is the prosthetic group. What is heme? Yes, beta. Iron.
Okay, sir.
Iron, it is the central part, uh, or the central element in case of hemoglobin,
Yes, sir.
right? So this hemoglobin, it contains a prosthetic group, a non-protein part that is called iron. Right? So this is how, and there will be a structure it will form here, like you can draw it to get an idea about this. Like for example, there's one tertiary structure, then you would have another tertiary structure like here, then you would have another tertiary structure like here, then you have another tertiary structure like here. So this is how you will get a structure, a three-dimensional structure that is called
Okay.
globular structure, and it is the structure of hemoglobin. All
Yes.
right. So in case of hemoglobin, it is a soluble protein. Hemo Hemoglobin. It is soluble protein.
Yes, sir.
All right. So
Yes.
it has 4 chains. There will be 2 alpha chains
Okay, sir.
and 2 beta chains.
Okay, sir.
Right.
Yes.
So this is how it forms a globular protein. Its hydrophobic side chain point inwards to the center of the molecule, and the hydrophilic side chain face outwards, making it soluble in water. As we know, It's the nature of every protein or every substance that may be hydrophilic or hydrophobic.
Yes, sir.
So as we know, as we have studied in case of the structure of cell membrane, there the soluble part of the membrane or the you can say hydrophilic part of the membrane that is towards water or outside, right?
Yes,
So it is this hemoglobin is composed of four chains, right?
yes, sir.
So there would be two types or two sides. One is hydrophobic, another one is
Look.
hydrophilic of this hemoglobin. And the hydrophobic side will always try to avoid water, and that will get into or close into the center,
o-
right? It means the central part of the hemoglobin that is hydrophobic, and because that is water heading, so it is getting into or enclosed in the center, not to expose to water,
Okay, sir.
right? So in case of hydrophilic, the outer structure of this hemoglobin that I have shown you above, so that structure which is outside, that is hydrophilic, right?
Okay.
So we can, I mean, uh, for for instance, we can categorize this structure like here. If this is a structure, so here you can this outer side is what? This outer side is hydrophilic, and the inner side, this inner part is this is what? This is hydro.
Phobic.
This is hydrophobic, and this structure is hydrophilic.
Philic.
All right.
Yes, sir.
This is our now. That's why it is soluble, because the outer side is very much exposed to water. So a mutation which causes one of the hydrophilic amino acids to be replaced by the hydrophobic amino acids, reducing its solubility, is responsible for a disease. We will study about this disease, and it may be asked you. It's important disease. Sometimes they may ask you separately, what is sickle cell anemia, or what is sickle cell?
Okay, sir.
Sickle sickle cell. Normally, red blood cells have very globular structure, because red blood cells are always rounded in shape,
Yes, sir.
right? And the hemoglobin which is present in it, that is globular protein.
Yes.
So, but if there would be any mutation there,
Yes, sir.
because because of the change of one amino acid there, in case of this hemoglobin, so that form a structure which is called sickle. Sickle is actually a crescent shape. Crescent shape. This globular structure will change to form this sort of structure.
Okay,
It is
sir.
crescent shape, like moon, right? So when hemoglobin attains this type of structure after the mutation, it will not be able to perform its function. What is its function?
To
What is
trans
the function of hemoglobin?
blood,
What is the function of hemoglobin?
blood transmutation.
Uh, Oxygen transportation. It is present
Oxygenated.
in the, yes, it is present in the red blood cells, right?
Yes, sir.
It is present in the red blood cells,
Yes.
and as this these red blood cells, they transport or they move throughout the body. So wherever the oxygen is needed,
Yes, sir.
so that will be provided by this hemoglobin, because it has trapped the oxygen uh, which is coming from the lungs and to the respiratory process.
Yes, sir.
So if the shape of the structure of this hemoglobin will change, it will not be able to perform its function, and
Yes.
there would be a disease which is called sickle cell anemia.
Okay, sir.
What is anemia?
No idea, sir.
Anemia is a deficiency of RBCs, deficiency of red blood cells,
Okay, sir.
right? A patient who has this sort of disease, definitely it is most of the time it is genetic, because there is a change in the genes, or there is a change at the level of protein amino acids. So
Mm.
once the amino acids will change, so they will uh, cause a disease which is called sickle cell anemia. It means body may have red blood cells there, but those red blood cells will not perform their function, because they will deform, they will change their shape because of this hemoglobin. Because the primary function of the red blood cells is to transport oxygen with the help of hemoglobin. If
Yes,
the
sir.
hemoglobin is deformed, if the hemoglobin is not performing its function, so there is no mean, there is no benefit, there is no purpose of this red blood cell in case of in the body of an organism,
Yes,
right? So here we can say that this sickle cell anemia is actually the deficiency of RBCs, and it is formed. It is a disease which is caused in organism where there is a change in the structure. So what we want to learn here, what is our objective here? I want to show you here the protein specificity. Protein is very specific. As we know, enzymes are very specific. So enzymes are proteins.
yes,
They
sir.
are very specific. For example, if you will change a single amino acid, protein will not perform its function. If
Okay,
you will change shape, as I have told you, in case of sickle cell anemia, you have changed a single amino acid.
yes,
And
sir.
that amino acid, once it is changed, so it is not able to perform its function.
Okay, sir.
On the other side, on the other side, if you will change its shape, as I have told you in case of boiled egg, you will not reverse that boiled egg back into the original form of the protein by any way, by any method. You cannot reverse that process of boiling, right?
Yes, sir.
So it means you will change shape by any way, by increasing pH or by increasing temperature, so that will also not perform its function. So what we have learned here, that proteins are very much specific, right? So mutation of hemoglobin at the beta chain, remember it is a change or mutation at the level of beta chain, not in alpha, at alpha chain. If anybody asks you this question, that how sickle cell anemia is caused in an organism, so you can say hemoglobin has four chains, 2 alpha and 2 beta. So there is a change of single amino acid at the beta chain, because of which the globular structure of the hemoglobin is converted into a crescent shape, which will not be able to perform its function, right?
Yes, sir.
Are you getting this point?
Yes, sir.
Is it easier? I mean, you need to repeat it.
सर एक दफा रिपीट करते हैं.
What I said, how sickle cell anemia caused in an organism. We will start from the structure of hemoglobin. We will say there are four chains in the hemoglobin protein,
Yes, sir.
two alpha chains and two beta chains. So there is a mutation of single amino acid at the point of beta chain,
Okay, sir.
right? Because of which the globular structure of the hemoglobin will be converted into crescent shape,
Okay, sir.
right? And this hemoglobin will not be able to transport oxygen in the body. So
Yes, sir.
as a result, sickle cell anemia will be caused to a to a to a person in whom this mutation is taking place. So
Yes, sir.
RBCs are produced in stem cells of the
Yes.
bone marrow. Now, a question is, which amino acid is going to change here? That I think we should know about it. As up till now, I said that there is a change in the amino acid, a
Yes, sir.
single amino acid that is going to change, and that will cause the sickle cell anemia.
Yes.
So there is a amino acid. Its name is glutamic acid.
Okay, sir.
Glutamic acid. right?
Yes, sir.
Glutamic acid is actually going to replace by another amino acid that is called valine, right?
Yes, sir.
So how it is different? I have replaced. I'm using the word here, replaced.
Okay, sir.
This this glutamic acid, this glutamic acid was actually hydrophilic,
Yes, sir.
right? So
Yes.
once it is replaced by this valine is hydrophobic.
Yes, sir.
So as a result of this, the property or the function of the of the hemoglobin protein is going to change because of this single change here. It is a single mutation. We call it point mutation, right?
Yes, sir.
So it will change the shape of the hemoglobin, and what would be after that? That will insoluble,
Okay.
in that will be
Inside.
less soluble, or I should not say insoluble. I should say less soluble,
Okay, sir.
right? Less soluble. Got it? So
Yes, sir.
this is how this sickle cell anemia is caused. Now let's move on another protein, that is collagen protein. Collagen, and it is also very common protein, and it's present in the body of an organism or even in a cell. Collagen proteins performing very vital functions here as well. Previous protein was globular protein. Now we are going to study another type of protein, that is fibrous protein. So this
Okay.
collagen is actually a fibrous protein. There are two main types of proteins. One is globular proteins, and we have studied its example in the form of hemoglobin. So
Yes.
now we have the collagen protein, and this collagen protein is actually a fibrous protein. Fibrous protein,
Yes, sir.
right? Vidya? So it is already insoluble. Where it is present,
In the muscles?
this collagen, this collagen is present in bones. It is present in tendons.
Okay, sir.
It is present in tendons. It is present in hairs. Keratin protein, that is a fibrous protein.
Okay.
Nails, your the protein which is, for example, the protein that is in our hair, that is a keratin protein. That protein is a fibrous protein. Our
Okay.
nails have the fibrous protein because the specific name of this protein is uh, keratin protein. Why would I?
Okay, sir.
So you can observe that, for example, tendons, the most powerful and pure collagen, tendons, right?
Yes, sir.
So you can talk about its structure as well, the structure of collagen. It is triple helix, writing here. Structure of collagen. So the structure of collagen, it is a triple helix. Helix. Once we say helix, so there should be a specific structure that should come in your mind, right?
Yes, sir.
Three polypeptide chains which wrapped around each other like a rope, right?
Yes, sir.
Which held together by hydrogen bonds. Hydrogen bondings are also present here, and they form a triple helix. I mean, about 1,000 amino acids they may present in each chain, right?
Yes,
So
sir.
every third amino acid in each chain is glycine, the smallest amino acid that allow three helical strands, right? So triple helix compound is also called tropocollagen. We call it tropocollagen. But one thing you should keep in mind, or important thing that you should remember, that it is a triple helix, so we can combine these triple helix together. For instance, that you have this one. There is a rope, like we are making a rope by combining two to three helix sort of like this, right?
Yes.
So they were combined with one another through they will combine with one another through covalent bond,
Yes, sir.
and they will also combine by the with the help of hydrogen bonding as well, right?
Okay.
So hydrogen bonds are also present here.
Okay, sir.
The two helix strands, for instance, they will combine directly with one another through covalent bond. But this hydrogen bonding is present everywhere where it is possible to make it more strengthened, right?
Okay, sir.
So this is how this collagen is present. So three helix, they wind together to form a collagen molecule, and triple helices held together to form a collagen fibrils. Collagen molecule is formed how? When three helix, when they combine with one another, they form like this, like this. You can add here more. This is what? This is a collagen molecule.
Yes, sir.
When these three, it is one, so you can add two more here. And when you will add two more collagen molecules here, they will form a fibrils. That is called collagen fiber. This is collagen fiber, right? So this is a second type of protein which is called collagen. Here you should keep in mind its function, and we will talk about its structure, like how its structure is. Definitely it is a fiber. So this fiber is elongated structure, and elongated structure in case of proteins are a helix. We have studied about the helices. So these helices are actually the helices, they are combined with one another to form a collagen fiber, right? All right, beta. Or is
Yes, sir.
okay. So then you will have we will talk about one thing or two points more. One is I hope water. We should know about this, the structure of water and the properties of water. You can talk about it. Yes, I think we are going to complete. And there is one more concept, and after that, that would be adhesion. What is adhesion? So it is also, I think, it's easy. So difference between hemoglobin and collagen, you you should know. In case of hemoglobin, what is the first difference? What is the structure? That is globular. Collagen is what? It is fibrous.
Yes, sir.
Hemoglobin is soluble or insoluble?
Global.
Soluble. And fibers are the collagens are insoluble,
Global.
right?
Yes, sir.
So what is the function of hemoglobin? To transport oxygen,
Precision.
right? Where it is present?
And
Red blood cells. Red blood cells.
yes, sir.
In red blood cells, right? Where is collagen? What is the structure of collagen? Triple helical structure.
Yes, sir.
And where it is located? Where it is present?
In the bone
In
standards.
the bones. Yes, in the bones and tendons and some other structures,
Yes,
right?
sir.
So you should know the difference between hemoglobin and collagen proteins, how they are different from each other, as their structure is different. Although the amino acids are the building block is same is not in the sense that every amino acid is a sequence is same. Their sequence may change, but they have the amino acids. And because of their change in the shape, they have different properties as well. So I think we have 2 minutes more. Okay, I think we will discuss two things more for about 5 minutes in our next lecture, and then we'll continue our next topic as well, right? So I think we should conclude here, right, Erase? Hello, जी बेटा आवाज आ रही है आप बोलिए.
मुझे सर जी आवाज़ आ रही है.
ओके, वही मैं कह रहा हूँ ना कि हम फिर उसको नेक्स्ट इंशाल्लाह बस 2-3 मिनट का इस टॉपिक में बात रहती
यस
है.
सर.
दो पॉइंट्स सबसे और विल डिस्कस
यस सर. ओके
नेक्स्ट
सर.
राइट.
ओके सर.
ऑलराइट, तो ये मुझे सेंड कर देना बेटा.
मैं सर आज
ये
करता हूँ.
हाँ बेटा, देन यू विल फॉरगेट इट. ओके, और फिर वो
ठीक
एंड
सर.
में
हाँ.
बनती नहीं. हाँ, फिर वो
ओके.
मेरे लिए बर्डन हो जाता है. ओके ओके, सी यू.